Welcome to Chapter 14, Part 1A. Given that a majority of people who die in any given year in the United States and an increasing percentage of those who die around the world are involved with some sort of cardiovascular disorder, some sort of cardiovascular pathophysiology, this becomes one of the major chapters this semester in terms of not only benefit to you as a care provider, but also benefit to you as a parent, as a loved one, as a care provider informally, as well as professionally. We do have a laundry list of things that we're going to be dealing with in this chapter, and so this is the study guide, or at least the beginning of a study guide for Chapter 14. We're going to be going over the cellular anatomy, some of the basic anatomical components and flow pattern. We're going to cover excitation-contraction coupling that we will basically build on from last chapter. We will also talk about graded levels of cardiac contraction, action potentials, and refractory period, and the very, very important reason why we have a significant refractory period in cardiac muscle. And the short answer is arrhythmia, but we'll get there. We're also going to talk about action potentials because that becomes the basis for how we understand pharmacology in the heart. By understanding action potentials, you can understand what's going on inside the heart at any given time. Electrical coordination of cardiac contraction, that's the classic electrocardiogram stuff. That's really, really fun stuff. I think it's kind of fun to know about an electrocardiogram, but if you don't have a good basis up here, it'll make this stuff in cardiac contraction all that much harder. We are going to get into the mechanical events, obviously systole and diastole, types of contraction, ejection. Then we get a little more technical in here. Now, if you remember from last module where we had, some complex images and things to understand, our sort of capstone, if you will, in this chapter will be the Wiggers diagram. It's going to show you multiple events occurring simultaneously. So it's going to show you several graphs all on the same time axis, so the x-axis. Stroke volume, cardiac output, those are mainly just terms that we will cover in order to understand cardiology and things going down the line. Just basically some terminology there. Couple notes about this lecture today in this chapter. We are going to go through an overview of the cardiovascular system, the cardiac muscle and the heart itself, the heart as a pump, but what we are not going to cover today are the aspects of pressure, flow, volume, and resistance, which what I'm going to do is I'm going to push this to chapter 15, where we talk about the vascular side of the cardiovascular system. So I'm going to make much of this lecture today and then this week about the heart itself. And then we're going to put the heart into the context of the vascular system, and that's when we're going to get into pressure, volume, flow, and resistance. Just a little review here. Obviously, this is stuff we've known about for a long time, probably since early in grade school. What we're doing is we're delivering oxygen and nutrients to our tissues. Another way to put it is that the heart is involved fundamentally in sustaining diffusional gradients. That doesn't make sense, right? You think, well, it's pumping blood and that sort of a thing. Well, the reason it pumps the blood certainly is to transport things to the tissues, but it is just as important to sustain concentration gradients. So that movement of blood across the capillaries in the lung or through the tissues in the systemic vasculature is there to fundamentally sustain concentration gradients, so that oxygen and nutrients and water move into the cells and wastes and that sort of a thing move out of the cells. Yes, we're going to deliver, immune cells, we're going to deliver hormones and stored nutrients, but I do want you to keep thinking about those diffusion gradients. That's going to be an increasing motif that we're going to be dealing with throughout the rest of this semester. So add that to your pocket of understanding when it comes to what the heart is actually doing in the cardiovascular system. It's sustaining concentration gradients. Yes, it is moving materials as well, but we already knew that, right? Right? Just some anatomical overviews and some component overviews of the cardiovascular system. We have the heart. The heart is a four-chamber, one-way, or should be a four-chamber, one-way valve equipped pump. It does have a central septum that divides the right half. This is the right, even though it's on the left side of this picture. It's oriented to the patient. So this is the right side of the heart, much less muscularized because the right side system is a low pressure system. Then you have the left side, which delivers blood to the systemic circulation. Left or right is sending it to the pulmonary circulation. The left is sending it to the systemic circulation. So the left's much more muscular. It's a higher pressure system because it has a lot more vasculature to deliver blood to. We certainly have veins, arteries, and capillaries. Capillaries have both a venule and an arterial component to them. That's why we sort of break them out from traditional veins and arteries. Again, that's going to be chapter 15. We're going to spend a lot of time on that, or a fair amount of time on that. And again, as I mentioned a few seconds ago, we have pulmonary circulation. So we actually have two different vascular circuits. We have a low pressure circuit, the pulmonary circuit, and we have a high pressure circuit, which is the systemic circuit. Now, even though the pulmonary is a low pressure circuit, it still delivers the same volume, or should be delivering the same volume of blood as the systemic circuit. If these guys get out of phase in terms of how much blood they are delivering, we start to see fluid buildup either in the lungs, or in some cases we'll start to see fluid buildup in the systemic circulation. We often call it JVD as a classic example, jugular vein distension. That's where the left or the right is not keeping up with the left. We certainly have blood, and that's chapter 16, so I'm not going to spend a lot of time on blood, but in chapter 16 we are going to get into some of the components and the functions of those components. Chapter 16 is a relatively shorter chapter compared to 14. 14 and 12 are big chapters this unit. In this slide, we're going to talk a little bit about the development of the heart. The reason I do is twofold. One is one of the great gaps in the biology department as it currently stands, though Dr. Weber is going to make strides to change that, is we don't have a developmental biology class, and we should for medical purposes as much as anything, because altered development is one of the major pathophysiologic mechanisms in the body, never mind the heart. The second reason I talk about this is that the number one cause of neonatal death in the United States is due to cardiac abnormalities. That is not due to anyone's fault. Yes, abusing drugs, abusing alcohol can and does cause these sorts of defects, but the number one cause of neonatal mortality in the United States is a failure of function at the time of birth, and that often is due to either developmental defects or failure to function at the time that it is needed. So let's go through development just for a little bit. Early on in embryonic development, what we have going on is a specialization of the tissue. It starts off as basically one sort of single tube, if you will. And through cell-cell signaling, which we talked about cell signaling, these cells differentiate and they begin to fold this, they begin to create this fold, if you will. We get this loop and fold back and structure starting to form the chambers of the heart. Eventually, pretty soon, we do develop an internal septum, we develop these one-way valves, and what we have going on is we have a downward deflection of blood towards what we call the apex of the heart, and then we have an upward ejection. The blood, as it's ejected out of the ventricles, is actually going towards the head. And that in part is due to this sort of folding that's going on, that's going to create eventually this four-chambered one-way valve setup heart. But like I said, some of the most common disorders that we see neonatally at the time of birth or lead to neonatal death. And don't ever forget that kids are tough. They really will withstand things that a normal adult will not. And so it hits pediatric departments pretty hard when these kids succumb. But some of the common things that we see are what are called atresias. That is to say, during this development, you have the hookup, the chambers don't develop properly. This is an example of one. This is called patent ductus arteriosus. What's happening during development is that you have a special blood vessel that runs to the placenta, and the gas exchange is occurring in the placenta rather than the lung. So you have to have a special blood vessel called the ductus arteriosus that brings that oxygenated or delivers blood to the placenta and returns oxygenated blood to the heart. That ends up becoming, when you're born, that ductus arteriosus closes and it becomes a ligament. And the remains of that ligament are actually what is involved in your belly button. So there's a ligament underneath your belly button that was at one time what's called the ductus arteriosus. It should go away, but in some cases it remains, and what you get is you get this mixing of oxygenated and deoxygenated blood. And so the patient doesn't receive as much oxygen as they should be. And some other things out there, all sorts of fun things. Here's a list of all sorts of congenital heart disease types. And again, I'm not going to test you on this. I do this because I believe this class is larger than just the minimum, if you understand. Maybe it's because I'm affected a little bit. My first niece died just a few minutes after birth because her pulmonary arteries did not dilate properly. So what happens is during development, the pulmonary arteries stay constricted, and that forces the blood through the ductus arteriosus. Well, when the child was born, those vessels should dilate, and they didn't, and so she basically died of anoxia because of persistent pulmonary hypertension. So it is something to be aware of. I don't want to, those of you who will be mothers in the coming years, I don't want you to walk around feeling afraid or that you have to do something special to avoid this. Just understand that it will happen. We do see birth defects in somewhere around 6 to 9% of births, even in perfectly healthy pregnancies. And heart leads the way. And let's return back to the regularly scheduled lecture. Again, we have a two-circuit setup. We have returning blood that comes into the right atrium, delivered to the right ventricle, and then is sent to the through the pulmonary circuit for oxygenation. Again, that's a low-pressure circuit. It's about 1/3 or 1/2 the pressure of the left side. That oxygenated blood returns to the left atrium, the left ventricle, and then sent out via the aorta to the systemic circuit, which includes upwards up into the head or the upper extremities or into the lower extremities, digestive tract, and so forth. By the way, we have 2 of the portal systems. We had 3 portal systems that I mentioned. Starting in endocrinology, we had the portal system in the anterior pituitary. We also have a hepatic portal system. And then we also have a kidney portal system. So 2 of the 3 portal systems are depicted in this vascular overview. And again, understand what a portal system is. We have a capillary bed, we have a condensation, and we have a second capillary bed before returning. Normally what we have is a single capillary bed, and then we return. Single capillary bed, but in these 2 instances and in the anterior pituitary, we have 2 capillary beds in series. Great question right here. Again, to help refresh your understanding of what a portal system is. Just a little bit of anatomical orientation. The heart is slightly on the left side of what's called the thoracic cavity. That's why when you say the Pledge of Allegiance or listen to the national anthem, you put your hand left of center on your chest. That's because most people's hearts are found left of center. About 1 in 10,000 people, though, have a unique genetic mutation in a gene called sonic hedgehog. Those developmental biologists like to have a sense of humor. In any case, if you have a mutation in the sonic hedgehog gene, instead of your heart being aimed over here, it's actually aimed the other way. Your internal organs are a mirror image of themselves. And so everything is like it's as if you were looking at it in a mirror. It's normal function, but it's just mirror image. My sister tells me about the story about the time they had one of these patients show up in her hospital, and the entire staff came running down to see this because it is rare, but it's otherwise asymptomatic. It could be actually in you guys. Yeah. If you happen to notice your heartbeat on the right side, you may have a sonic hedgehog mutation. In any case, we do need to understand the base of the heart versus the apex. The base is actually closer to the head. It's where the outflowing vessels are found. What's happening when we constrict the ventricles, we're pumping upwards towards the head, we're pumping upwards towards the base. So we have the base at the top of the heart, and we have the apex at the point of the heart. And while the heart is mostly muscle, it does contain other types of tissue, certainly fat tissue, secretory epithelium. That what you call a pericardium is a secretory, three-layered secretory epithelium. Puts a little bit of fluid inside the pericardium in order to prevent irritation as the heart sort of squeezes, and it sort of squeezes and twists at the same time. The heart doesn't just squeeze, it actually sort of spins as it squeezes. And so having a little bit of lubrication in that pericardial sac is very, very important in order to prevent inflammation and irritation. So here's the pericardial sac. That pericardial sac is attached to the diaphragm to help kind of stabilize the heart a little bit. One of the things I noticed that is when people have repeat open heart surgeries, when they have a second open heart surgery, it's usually much more complicated because when they open up that pericardium, when it heals, it starts adhering to the surface of the heart, and it's much harder to extricate from the heart during the second open heart surgery. Another reason to avoid open heart surgeries in the first place. Again, here's the base up here in this part of the figure, and here is the apex down in this part of the figure. Again, some anatomy. If you've had 305, you're all set, but if you haven't had 305, you might want to just associate yourself with these different structures. The inflowing vena cava, right atrium, right ventricle, pulmonary artery, which is shown in blue because it is deoxygenated blood. Even though it's an artery, it's deoxygenated. It returns via the pulmonary vein, which we cannot see. It's behind the pulmonary artery, left atrium, left ventricle, and then the outflowing artery known as the aorta. And if you haven't had or if you haven't studied the cardiac circuit, I didn't believe it until I did actually have a student show up who said she didn't know the cardiac circuit. This is the sequence of events. Blood from the systemic vasculature arrives via the vena cava and moves downward through this progression. So if you haven't had the cardiac circuit or the cardiac sequence of events, then by all means study this slide. This figure here is an extension of the previous slide. However, now we're starting to go into the interior chambers, and we're starting to see the valves and some of the interior structures of the heart that are very, very important for one-way flow in the heart. Again, where we have blood returning from the vena cava to the right atrium then into the right ventricle. Now I want you to notice something about the interior surface of the ventricular chamber. I'm not sure so much about the the atrial chambers, but certainly the ventricular chambers, you have this sort of reticulated structure, including some special muscular structures called papillary muscles. These papillary muscles contract at the same time the rest of the myocardium contracts. What they're doing is they're pulling tension in order to retain patency of these arteriovenous valves. What we can get sometimes if you have pathophysiology of what are called the AV valves, the arterioventricular valves, you end up, instead of getting blood going the outflow way into the outflowing arteries, you end up pushing blood back into the atria, and that's called regurgitation. So we have the one-way valves here. They're called atrioventricular valves, or AV valves. Those valves need reinforcement while the ventricles are contracting. And so we have these papillary muscles and chordae tendineae, they're over here as well. And they are like cables on the Golden Gate Bridge. They pull tension in order to reinforce the rigidity of those valves so you're not accidentally pushing the valves back into the atria. That's called regurgitation. And I'll show you some more pictures of this. But we have blood coming through these AV valves, and then when the ventricles contract, we want the blood to go out the outflowing arteries, and so we have those contractions of these chordae, or these papillary muscles, holding tension on those valves so that they don't prolapse back into the atria. So the two sets of heart valves ensure one-way flow. We have the atrioventricular valves, abbreviated AV. They separate the atria and the ventricles. On the right side, it is a three-leaf valve, and so we call it a tricuspid valve on the right side. And on the left side, and this one is the big one, this is the one that we see most pathophysiology. This one has the most strain. It's called a bicuspid valve, or sometimes called a mitral valve, named after a bishop's mitre, which has kind of two leaves on it. That's on the left side, and this is by far the most common cause of valve pathophysiology. Regurgitation, valve failure is this bicuspid / mitral valve. The outflowing valves from the ventricle are called semilunar valves, and these separate the ventricles from the outflowing arteries. And the valves are named after the vessels to which they flow into. So on the left-hand side, we have the aortic valve going into the aorta from the left ventricle, and on the right-hand side, we have the pulmonary valve going to the pulmonary arteries. And again, as the ventricles contract, those papillary muscles tense up. Those valves will shut even though the papillary muscle is pulling on them. And that chordae tendineae there, that provides reinforcement of the valve to prevent prolapse of the valve, which is a problem that leads to persistent regurgitation, or regurgitation for any reason whatsoever. This is just a top view showing you here is the tricuspid. That's the right side. You can see the three leaves here. And here is the mitral / bicuspid valve on the left side. But again, the papillary muscles and the chordae tendineae reinforce the valve to prevent it from prolapsing or folding back into the atrium, or allowing for backwards flow. We call that regurgitation. All blood should be leaving through the semilunar valves, either into the pulmonary artery or the aorta. While during relaxation, the papillary muscles are relaxed, the tendineae are relaxed, the valve will allow for passive as well as active filling. I'll let you in on a little secret that most people don't realize until they take this class. 85 to 90% of the inflow of blood into the ventricle is passive. That is to say, when the atria squeeze, when they undergo atrial systole, they're only adding about 10 to 15, maybe 20% of the overall volume to the ventricle. They are not adding a majority of blood. They're only sort of preloading the ventricles. And we'll talk about that. It has to do with link tension relationship. They're actually adding a little bit of extra blood in order to stretch that ventricle a little bit so you get a nice, solid ventricular contraction. So 85 to 90% of the blood fills passively during what we call diastole. So when the ventricle is relaxed. And again, here's the valve open going into the right side. It's a three-leaf situation, two-leaf situation on the left. But again, this happens during late diastole that we get most of the filling in the ventricles. Late diastole meaning that the atria and the ventricles are relaxed. 2/3 of your life you're in late diastole. So for every hour, 40 minutes, your heart is not pumping. Your heart is in relaxation. It's kind of a strange thought. 18 out of 24 hours, or no, yeah, 18 out of 24 hours, your heart doesn't beat. Couple of nice videos to sort of help you see this visually, and it really does help to see this visually and then go back to the slides if it's giving you trouble. Some of the things that I've noticed on the exams coming up the exams that we've had in the past in this class, is that it seems like you are not able to visualize some of this stuff. So I want to do as much as I can to help you see this visually. And so use these videos. They're only a minute or two long. Worth your time. Go over and watch them. Same thing's true. If this lecture is not helping you to grasp things visually, by all means, Crash Course, Khan Academy, Draw It to Know It are all other options. I would do anything that I could to be a better lecturer. I'd be honest with you. I would do backflips if I thought it would help. But if my lectures aren't serving the purpose, then by all means use other resources. Good students do that. One thing about cardiac muscle that shares a characteristic with smooth muscle is that cardiac muscle will and does receive calcium from other cells, or in some cases from extracellular fluid. That's because cardiac muscle is involved or can be stimulated by hormones, in the case adrenergic or hormones, or it can be influenced to contract through gap junctions from calcium from the cytosol of other cells. So it can initiate an action potential just like a skeletal muscle, but it can also be initiated to contract from extracellular sources of calcium. Now, cardiac muscle is a lot more like skeletal muscle. It doesn't use calmodulin like our smooth muscle does. It uses troponin, tropomyosin like the skeletal muscle. But it is an in-between in terms of the initiation, the excitation-contraction coupling between smooth muscle and skeletal muscle. The myocardial part of cardiac muscle, the muscle in cardiac muscle, is actually composed of two different cell types. We have what are called contractile cells. That's illustrated in the next figure here. When you see this number 3, I'll show you this in just a second. Those are the true striated fibers. They have regimented sarcomeres. They have the intercalated discs that we see. They're mononuclear as opposed to polynuclear. So skeletal muscles are polynuclear. Smooth muscles and cardiac muscles are mononuclear. Yeah, here in number 3, here's what I'm talking about. This is a myocardial contractile cell. Now, number 2 here, this one right here, I've got that highlighted there. These cells are also myocardial muscles fibers, but they are autorhythmic muscle fibers. We call them pacemakers, and we find them throughout the myocardium, not just in nodes or in the autorhythmic tissue. All myocardium is autorhythmic. It has the ability to self-sustain contraction rhythmicity. But we do have these pacemaker cells, again highlighted here, number 2, and in here probably number 1 as well. I don't, not sure what's going on there, but this is certainly looks a lot like an autorhythmic cell. These guys are the ones that initiate the signal for contraction under normal circumstances. If for some reason these cells start malfunctioning or stop functioning altogether, we do see autorhythmicity in the contractile cells. They have the ability to just spontaneously depolarize. Okay. There's the autorhythmic cells are smaller and fewer than compared to the contractile cells, but they do not have organized sarcomeres. You don't see the striations in these cells. That's because their job is to initiate action potentials and or calcium signaling into the contractile cells. As I've mentioned in the previous slide, the cardiac muscle cells versus the skeletal muscle, they are smaller because they don't fuse like skeletal muscle precursor cells do in order to create skeletal muscle fibers. So they're smaller, they're mononuclear. Skeletal muscle fibers are very, very large, relatively speaking, and polynuclear. Cardiac muscle contractile cells are often branched. That is to say, they branch, and so they aren't just a single tube, sort of like skeletal muscles are. And they have these things called intercalated discs, which are full of very, very durable junctions, cell-cell junctions. We call these things desmosomes. They're like tight junctions, but they don't behave like tight junctions in terms of the ability to regulate paracellular movement. Tight junctions are a lot like desmosomes, but they regulate paracellular movement. But desmosomes don't do that. They are very, very strong, and I'm interlinking my fingers here. I wish you could see that, because they're allowing for force transfer. Then in those intercalated discs, you also have numerous gap junctions that provide, it says electrical connection, and that's true, but what we're really talking about are calcium, perhaps potassium as well, but calcium's the big one. Calcium's the big one going through these gap junctions. There are more T-tubules. That's because we are more dependent on synchronous contraction in the heart than perhaps the skeletal muscle. Yes, if a skeletal muscle contracts asymmetrically, it's painful, but it is not a potentially life-threatening situation. So we have a lot more T-tubules. That's because we very much need to have unified contraction in the cardiac contractile fibers. The sarcoplasmic reticulum is smaller. It doesn't hold quite as much calcium. That's because we're depending on more calcium coming from extracellular sources, either other myocardial cells or from the autorhythmic cells or straight from the extracellular fluid. That's part of why we see influence of cardiac contraction from hormones, because it will alter the calcium kinetics. It will let more calcium in from extracellular sources. One thing we cannot tolerate in cardiac muscle is fatigue. And so we have tons and tons of mitochondria. It's almost like these guys are bags of mitochondria, because 1/3 of the cell volume fully is mitochondria. Because we need ATP, we have to have it. If we don't contract our myocardium, if we fatigue, we call that heart failure. And heart failure is not a good thing for any patient, any level of heart failure. And there's just a summarizing picture showing you the histology here, the branched myocardial contractile cells. Here's one of these autorhythmic cells, probably one of these guys here, and that sort of a thing. And the intercalated discs, which have desmosomes for transfer of force and gap junctions for transfer of ions, particularly calcium, but I'm going to guess probably potassium as well. And so the last few slides in this part of the lecture, we're going to cover the big mechanism in cardiac muscle, and that is excitation-contraction. Now you'll notice this isn't cardiac, this is skeletal right here, because this is our base model. What I'm going to do is I'm going to compare and contrast the cardiac muscle to the skeletal. So ahead of number 3 here, 1 and 2, what you have going on is you have the motor end plate, you have secretion of acetylcholine. That triggers a sodium influx and an action potential. That action potential spreads down the T-tubules. That T-tubule then, or that action potential then causes a voltage-mediated change in the shape of the DHP receptor, dihydropyridine, DHP. DHP is physically connected to the ryanodine receptor, RYR, and when it changes shape, it pulls open that channel. The calcium floods out of the sarcoplasmic reticulum, interacts with troponin, tropomyosin, and we get that power cycle that we talked about with myosin. We go from the relaxed state, we flex the myosin, we lose the inorganic phosphate, we lose the ADP, we're in the rigor state, more ATP causes the myosin head to release and cock again and repeat, so long as calcium is present. Now let's turn to the cardiac muscle. So we get some sort of an action potential that can enter from an adjacent cell through gap junctions, or it can be initiated by a voltage-mediated event. In any case, we get an action potential on the surface of the cardiac contractile cell. That will then spread down through the T-tubules, and instead of encountering a voltage-gated receptor called the dihydropyridine receptor, instead, we are going to get a voltage-mediated event on what's called an L-type calcium channel. That L-type calcium channel is a voltage-gated channel. L-type means that it is voltage-gated in the span of things, because there's a whole bunch of calcium channels out there. In any case, the voltage causes that L-type channel to open, and calcium from the extracellular fluid moves into the cytoplasm, where you notice that the L-type calcium channel is not connected. It is not physically connected to the cardiac RYR. The cardiac ryanodine receptor is not physically interacting, unlike the skeletal muscle. It turns out that the ryanodine receptor in cardiac muscle is a calcium-sensitive calcium channel. It is a calcium-gated calcium channel. Write that down. It is calcium-gated calcium channel. The ryanodine receptor opens in response to calcium levels rising in the cytosol. That in turn releases more calcium, so we get a sort of positive feedback situation going on. A little bit of calcium comes in, more calcium is released, and we can actually detect that using special calcium-sensitive dyes. All those that influx of calcium, both from the L-type calcium channel and from the RYR, a spark. That's because when you look at it microscopically, it looks like little sparks. And I have a video for you guys to watch so you can see that. If enough sparks ensue, we move on to a large wave, if you will. That large calcium wave then is as a summation of these sparks. It causes a positive reinforcement. So we get a positive. More calcium causes more RYRs to open, more calcium, more RYRs, more calcium, more RYRs, and we get this large calcium wave inside the cytoplasm of the contractile cell. And that's what's shown in this video. I can't include it as part of the video I'm making for you. But what you see going on initially is you see all these little flashes over here, and soon enough of them happen, and you get this huge wave of calcium that spreads down the cell like this. It's a positive reinforcing phenomenon. And then you see the wave go away almost immediately. That calcium comes and then it goes in a split second. Comes and goes in a split second. So this video is worth your time. Just watch it for a minute and you can see what we've just been talking about in the previous few slides. So we get that calcium wave going on, and that calcium signal then initiates the contraction pattern in the myosin, just like the sarcoplasm, or just like the muscle fibers in skeletal muscle. You move tropomyosin, that causes a tight interaction of the myosin with the actin, and we get the same circular-like progression of contraction that we get in the skeletal muscle. However, the kinetics of removal of calcium from the cardiac muscle are much more robust than in the skeletal muscle. That's because generally we want a single contraction and a relaxation, sort of like a twitch. One contraction, one relaxation. We don't want multiple twitches. You know what we call multiple twitches in cardiac muscle? Fibrillation. And you've probably heard that term before, and it's not a good term. So we want a single contraction relaxation cycle to occur in this muscles. So what happens is as we contract, the myocardial contractile fiber is actively removing that calcium as quick as it can. And so let's get into the mechanisms of how we get rid of calcium from the cytosol of the contractile fiber. One is just like the skeletal muscle. We have a calcium ATPase, primary active transport. So this protein hydrolyzes ATP as it moves calcium from the cytosol back into the sarcoplasmic reticulum. But that's not the end of the story. As I mentioned in the previous slides, cardiac muscle is more robust in its removal of calcium because we want a single contraction relaxation cycle. Step 9 is secondary active transport. Because calcium is higher outside the cell, it will not diffuse on its own. And so what do? We use the sodium gradient across the plasma membrane, back to our sodium-potassium ATPase, and we use this NCX, which stands for sodium-calcium antiporter, the X meaning antiporter. So we use secondary active transport. We're not hydrolyzing ATP here. We're hydrolyzing ATP in the sodium-potassium ATPase, generating a sodium gradient that we then allow sodium in to force calcium against its concentration gradient. So we have primary active transport here at 8. We have secondary active transport of calcium out of the cytosol in 9. And so that is secondary active transport, the sodium-potassium ATPase antiporter, which they often use X for antiporter. And so, like skeletal muscle, I want you to understand where the major areas of ATP hydrolysis are occurring, sort of the big reasons we have ATP in cardiac muscle. We certainly have ATP hydrolysis here in order to sustain the sodium-potassium gradient. That's not only important for getting rid of calcium, it's also important for the action potentials on the surface of this cell. We have ATP right here at 8. That's the calcium ATPase. It's a uniporter. It is pumping calcium back into the sarcoplasmic reticulum out of the cytosol. And then we have ATP down here, ATP hydrolysis that cocks the myosin. It doesn't cause flex, it causes cocking. We're transferring the energy from the phosphate to the protein structure of the myosin. And then when the myosin flexes, that's the release of that potential energy that's stored in the protein, sort of like a spring, if you will. And certainly ATP is used for other things, but this is the big three. Remember, we talked about ATP hydrolysis in skeletal muscle. We need to know about ATP hydrolysis in cardiac as well. That's it for today. Thank you for your time. I really appreciate it. Let me know if you have any questions. I've created a chat forum on ACE. I'm also going to have my regular office hours from 2 to 3:30 using Google Meet. I'll be publishing that link for you guys, as well as from 7 to 9 p.m. on Mondays. And again, thank you. Hang in there. Hopefully you have fewer distractions now, as opposed to our normal life. I don't want to coop you guys up, but use this as an opportunity to do really good work. Many of you are better students than what your scores are indicating. I think this is a good time for you to really hone in on those skill sets. So let's do this together. And again, thank you. I will be talking at you soon enough.